Welcome. Now we're going to look at meiosis. Meiosis is the production of sex cells, the sperm and the egg. Now this is vital in sexual reproduction, and we're going to talk about how that happens in the cellular level. Now obviously we've already talked about mitosis and how it produces body cells, and how the first cell, if it has 4 chromosomes, will go through mitosis one time and form 2 new cells, and each cell has 4 chromosomes. Meiosis is how we cut those chromosomes in half in order to have sexual reproduction. Because in order to have a cell with 4 chromosomes, we have to have a cell with 2 chromosomes in each.
So meiosis is a process of reduction division in which the number of chromosomes per cell is cut in half through the separation of homologous chromosomes in a diploid cell. That was a lot of big words in there. Now what does diploid mean? Diploid means 2 sets of chromosomes, and haploid means 1 set of chromosomes. Homologous means chromosomes that each have corresponding chromosomes from the opposite sex parent, the male and the female. Now, what are the pairs? Now we have these things called pairs. In the human we have 23 pairs, and if you take 23 pairs, that means you have 46 chromosomes. Now each one of these pairs has very different DNA and traits on them, genes and genetic code that carries who you are and what you are. The interesting thing about these pairs is, notice in the diagram here, we see that so far in mitosis what we've seen is you see one chromatid makes a copy of the second chromatid. Well, that's going to happen in meiosis as well, but the only difference is they will switch it up. And this is what allows for variation. So they will get right next to another and they will switch genetic coding. That way they can actually switch things up and variation is possible. The meiosis stages usually involves 2 distinct stages, meiosis I and meiosis II.
The cell prepares for meiosis just as it does for mitosis. But as the DNA condenses, each chromosome finds its homologous counterpart. These matching pairs of chromosomes hold equivalent genetic information, 1 set from mother and 1 from father. Next comes the first shuffle of nature's genetic deck. The 2 homologs trade genes in a process called crossing over. This forms new hybrid chromosomes. The spindles then place maternal and paternal chromosomes randomly on each side of midline. It is another shuffle that can lead to a host of different genetic outcomes. Finally, spindle fibers pull the homologs apart. This leaves the sister chromatids intact. With twice the amount of genetic information the cells will need, nature must cut the genetic deck in 2. The second division produces 4 unique sets of chromosomes. Because they carry half the genetic information, we call these haploid cells. Many of them will mature into sperm and eggs, the reproductive cells called gametes.
Again, let's go ahead and go through each one of the phases. And again, in meiosis, we give a number 1 and number 2 after each phase. That way, you kind of get an idea that it separates meiosis from mitosis. So, prophase I is in meiosis. This is where all the chromosomes are going to come together and they're going to start getting to that point where the nuclear membrane dissolves just like it does in mitosis. The only difference is that the crossover effect may actually happen. And that allows for, again, like we talked about before, variety. Here we see in prophase, the chromatids have made copies. We do have a crossover coming into effect in two different spots here. Again, if you take a look at this cell, how many original chromosomes are in there? If you answered 4, you're correct. 1, 2, 3, 4. Now, the problem is in meiosis, you don't normally have original chromosomes after this stage because of the crossover effect, and it allows for variety. Notice the nuclear membrane is disappearing. The spindles are going to the opposite ends. Metaphase I. Now this is the big difference here. The chromatids do not line up on the equator, they line up at the equator. So we see in metaphase I that they're going to line up in pairs. So they're going to line up in different alignment. They're not going to line up exactly on because we're not breaking the centromere. We're not breaking the center of this. We're just pulling the pairs apart. Now in anaphase, obviously, it's just like all the other anaphases, and it pulls the chromosomes apart. Now we see here, we see a pair here as well, a pair here as well. Now, the only difference is they are different because of the hybrid that has been created through the crossover. Now, telophase I and cytokinesis steps in. 2 cells are now formed. Now the 2 new cells will go through meiosis II or prophase II, and now we are going to cut this in half. So what's going to happen is the nuclear membrane is going to dissolve once more. The spindle fibers show. Now the chromatids, they don't need to copy anymore. They're not copying because they've already been copied. Now what they're going to do is they're going to go to metaphase II and line up on the equator just like they did in metaphase of mitosis. Now we see the anaphase kick in and the splitting of the two, and we see half the original. So here we have anaphase I showing that it's pulling the pair apart, and this is anaphase II where it's pulling each individual chromatid apart. And what's fascinating and cool about this is now we have 4 cells that are all different. This cell is totally different than this one, totally different than this one, and totally different than this one. However, on this one over here that we see in the far right, there are going to be some similarities between the one here next to it. There's going to be some similarities because they do hold some of the same genetic code, but there are some variety and differences. And that's why when you take a look at brothers and sisters, you can see that you have some traits that are similar to your brother or sister, or, as a matter of fact, some of you may not. In males, meiosis results in sperm cells. In females, meiosis results in egg cells. Again, this is just kind of a breakdown between the two of mitosis. Results, basically, you get 2 diploid cells, which means you get 2 of the same exact cells when you go through mitosis. In meiosis, you get 4 haploid cells, which means you get half the original number of chromosomes, but you get 4 cells. In mitosis, the cells are identical, and then in meiosis, they are different. And of course, mitosis, we've talked about before, are the body cells, and then meiosis are the sex cells.
And that's it for meiosis. Meiosis is the sex cell reproduction. It is the way that we're able to have male and female produce an egg and sperm and bring those together to form a new individual of the same species. Without that, we wouldn't be able to. So, if you have any further questions, make sure you keep going over meiosis, keep studying, keep looking at it. Now, just like we did in mitosis, let's take an example. If I start a cell with 4 chromosomes, it goes through meiosis, how many cells do I end up with? If you said 4, that is correct. how many chromosomes are in each cell. If you said 2, you're correct, and you're on your way.